Thank you. Good afternoon, everybody. When we think about the future of health and personalized medicine, we don't usually think of proteomics as being at the forefront of that frontier. The purpose of my presentation this afternoon is to make the assertion that that's wrong, and actually proteomics could be the most important omic as we go forward. But let's start at the beginning. What are we trying to achieve? And let's look at the right-hand side here. And what is a kind of supply chain diagram to get there? So what we really want is lower cost, better outcomes, and improved experiences. But let's start on the left. If we had more insights, that is, better measures of current state and future trajectory on the course of wellness towards illness, then maybe we'd be able to match those things to actions. And if we matched actions to them, maybe they would have clinical impact and we'd be able to prevent some of those bad things from happening. And if we did that, we'd get some elements of the quadruple aim. So in thinking about this pathway then, let's start at the beginning and let's think about, well, where did these wonderful insights actually come from? The traditional view has been to come up with a wonderful causal theory that maybe is based on decades of laboratory experimentation, and to take an assay for that measure forward, one diagnostic measure through various phases of development all the way through to commercial assay validation. There are some issues, though, that if you think about what weaknesses and issues there might be with this, that the best example in our history, I think, is HIV viral load, because HIV is pretty close to being a unidimensional disease. And if we measure the viral load, then this captures about 80% of the outcomes. Of course, the bit it doesn't catch and doesn't explain is why do people with low viral loads still die of cardiovascular disease? But other measures that we revere equally highly are actually not so good. That the original causal theory for LDL cholesterol is it's blocking up our arteries. Well, that was the issue that was the theory when I was at medical school. And we all know that's way too simplistic, and we all know that there are lots of events despite good control of LDL. And as for diabetic control being measured by HbA1c, it's got a very poor association with outcomes. So this, as a high throughput way of getting new health insights, doesn't cut it. And the reason it doesn't is this. That if you take any disease, and let's take diabetes as our example, and you say, is there such a thing as a simple diabetic that I could use to train my health insight model on, you can actually see that there's no space in the middle where someone is just a simple diabetic and doesn't have one or more of the comorbidities. So if people and diseases themselves, even within the diabetes paradigm, there are subphenotypes. If all of these things are driving towards multidimensional needs, then that means we need a multidimensional discovery and validation program. So how do you do that? Well, oftentimes you do something like this step diagram, that because discovery technologies measuring lots of dimensions are expensive, measurements are expensive and more measurements are more expensive, you tend to do discovery in limited numbers of subjects. Maybe you even enrich this for likely candidates from the literature. And then the object is to get down to a small number of measurements that can be put onto the FDA-validated platforms at low cost. The issue here, of course, is that you're actually, in driving down to a high-scale technology for delivery, you're actually losing information. But you get the low-cost delivery, and you get, to some extent, a broader discovery. But this path is difficult. You lose information. You have to keep switching technologies, platforms. And once you've got here, you've fixed it forever. You can't change it. You'd have to go back to the beginning to enhance your offering. The next way of thinking about where health insights might come from is, well, let's have the discovery technology and the delivery technology be the same and highly multidimensional. And let's do discovery in thousands of people, and let's measure everything such that we can give lots of outputs. So this would seem to be the ideal, but it's actually very demanding. So if you do this, you don't lose any information. Because you're always measuring everything, you can upgrade your algorithms, and it fits the multidimensionality of people and diseases. So that all sounds great. The trouble is that the cons are also substantial as well. If you're doing this, you have to be sure that the measurement you're making is the information you're looking for is actually encoded in the measurement that you're making. It also demands low cost and high precision in that if you're going to use this for medical information delivery, it can't be thousands of dollars a pop, and it can't take 3 weeks to run. And this also demands very large data sets because you're trying to characterize multiple subphenotypes of disease and multiple environmental and genetic variations in the people in the measurements you're making. So let's look at how we might overcome some of the cons to enable this idealized multidimensional discovery and delivery. So this is a graph that on the right-hand side about the performance of a set of tests for diagnosing NASH, which is a disease of obesity. It's inflammation and fibrosis of the liver. And for those of you who are not familiar with these rather confusing charts, where you want to be is up in the top left-hand corner, which is a sensitivity of 100% and a specificity of 100%. What that means is everybody who has the disease, you find them, and everybody who does not have the disease, you correctly classify them as not having the disease. And you'll see in this set of curves that when we did an experiment with proteins, we got this upper curve, which is about 90% area under the curve. Big data from electronic health records was about halfway in between, and a genetic marker was down low. So, the one point is, well, this is about where is the information encoded. And in this case, it turned out that more information was encoded within proteins than it was in the electronic health records. But the other point I'd like to make is this diagonal line here is zero information. It's like a coin flip. So heads or tails would be 50-50. Simple heads you have disease, tails you don't. If you want to get more sensitivity, you can make it easier to diagnose, and you could say, I'm going to flip a coin 10 times, and if any one of them is a heads, you've got the disease. So the sensitivity goes way up. You correctly find nearly everybody with disease, but you also falsely classify nearly everybody who didn't have the disease. So you can go to the other extreme and say, I'm going to flip a coin 10 times, and if they're all heads, then you've got disease. You make it very difficult to diagnose. So then you get great specificity because you correctly diagnose people who didn't have the disease as not having it, but you've lost nearly all your sensitivity. Now, why am I talking about coin flips? Well, because I could make a warehouse full of robotic coin flippers, and I could generate enormous amounts of data. Or I could create an app that simulated coin flipping. And again, I could generate enormous amounts of data. But the point is, there would be no information in it. So when we talk about big data, we're not really talking about big data. What we really want is big information. And the point of the coin flip line is they're not synonymous. The other thing is that the most important omic is actually not proteomics, it's economics. And the middle line, even though you got modest performance out of the electronic health records, the extremely low acquisition costs coupled with modest performance might mean that that's a reasonable thing to do. The problem with the proteomics has been, yes, you get more information, but you've got more cost as well. So it's the balance between the cost of acquisition of the information and the information content that you have to manage. So one of the points is, can you actually get more information, and can you get the costs down of acquiring it? And one way we've been doing this is to take this SomaScan assay and to try and get the costs down, the precision up. The latest version of our assay, which measures 4,993 proteins, I've just rounded it up slightly, started running it last week, full scale after clear validation, which means we're already 2,000 samples in. What it does is it uses a DNA reagent to find the shape of an epitope on a protein. So it's measuring the availability of that epitope, which can change as the protein concentration goes up and down. but it can also change if the shape or charge on that protein goes up, it changes. Or it can change if there's something else occupying that binding site. So it's sensitive to 3 different kinds of biology. And it's precise, and our intent is to use it for discovery and delivery, same assay, no technology change. So how does this apply to scale? So I put this slide together, and in one way, it's a boring slide because it's kind of boring format. But in the other way, I looked at it, and that was actually pretty shocked by what we've put together. This is big data. The previous world record for a single protein study about 3 years ago, we held it at about 2.7 million measurements. And then our collaborators in Cambridge University beat it with about 5 million. And then a big pharma company we collaborate with has beat it with about 25 million. But within the next 18 months, within the same database, we'll actually have 1 billion protein measurements. And they will have over 1 million participant years worth of follow-up. So people have looked at this and said, well, why do you need so much data? And I think it's about the respect for the need for personalized medicine. If you believe that individuals themselves are genetically and environmentally different, and if you believe that diseases are heterogeneous, then when you use machine learning to find the optimal combination of measures that reflect those things, you need to make sure that every combination of subphenotype and genetic and environmental variation is represented within your data set, because that's where the information is. So that's really what's driving the size of these things. And what will this enable us to do in the future? Well, remember, this is discovery and delivery. So we can take a person and we can have proteomic surrogates for a lot of different health issues all at the same time. And to go into a little bit more detail, this enables you to say, is this person facing a health issue? Let's say they have diabetes. And therefore, which insights can I package together for them? Of course, you could have an all-you-can-eat buffet. We're currently working on about 64 different algorithms, surrogates of current state or future state. You could give everybody all 64 and let them choose, but I think that would be overwhelming. The path we're taking to start with is to create bundles of validated insights that match a person's health issue. But this shows you that because you always measure everything, you can actually repackage these things to fit a different health situation, a different decision. And it's really all about repackaging bits of software, that all the measurements stay the same, and each algorithm is a piece of software that you can actually change, enhance, join up with other measures. So what evidence have we got that this actually might work, this approach to using machine learning, a big discovery platform? What evidence have we got that this is actually better than existing alternatives? The first one came a couple of years ago with our approach to predicting cardiovascular events 5 years in the future. And what you're looking at on the right is a calibration plot. This one's for about 1,000 people. So what we do is to run the risk algorithm on 1,000 people, rank them from worst to best, and then split them into deciles. So each of these bars is about 100 people. And then there's 2 key things you compare. One is, if you make a prediction, how well did it match the actual observed event rate in that group? And you can see that the bars match very well. And the other thing you look at is how different were the worst and the best. And you can see that they're pretty different. In fact, when you compare them with the best available alternatives, this dynamic range is about twice as good as the best combination of risk factors and laboratory measurements, and about 7 times as good as the best available combination of genetic markers. So this is a bit like the liver thing, that there is information encoded in proteins. The other interesting thing here is that we gave the machine learning algorithm not just proteins, but all the available data to choose from. It did not include LDL cholesterol or age or sex in the model at all. It only actually chose proteins. Again, in this condition, the proteins that are encoding for more of the information than the other things that we've traditionally used. The next thing that I'll give you to provide some proof of concept is about measuring change. Well, a lot of the things we use in medicine today, a lot of the ways we predict the future risk are things that actually we can't change. In cardiovascular disease, it's often age and sex, and they're quite difficult to change. Anyway, in some directions. This is a study where we applied that same risk algorithm in a big study that Pfizer did from 2007. In 2007, they stopped this study because people were dying on the new drug, which was called torcetrapib. And they stopped it at 18 months. And what we had here was they had samples at baseline that they'd stored in the freezer, and they had samples at 3 months in. And we ran those samples and applied the risk score to them, and you can see that in the group that just had their dose of atorvastatin optimized, we measured the cardiovascular risk to go down. And in the group that had torcetrapib added to their atorvastatin, we saw the risk go up. And the interesting thing here is that the difference across these groups that we measured or predicted was actually within 0.1% of the actual observed event rate. That was at 18 months, and whereas this was done at 3 months. In contrast, the risk factors, LDL cholesterol, if you do a Framingham risk score on these people, they all improved in the torcetrapib group, which was why the drug was being developed. So they were actually false positives. So those are two key features of measuring health status is, well, can you predict the future? And if you do something to change that future that would change an outcome, can you measure it before it happens? And this was proof of concept that we could do that. So I think we've got a decent case for having the substrate for doing this. We've got lots of good, well-labeled sample sets. The other thing I should mention about the sample sets is what they reveal about me is I'm impatient, and that we've got 1 million patient years worth of follow-up because we've used biobank sample sets that already have 10 years or 20 years worth of follow-up. Another advantage of using those sample sets is that the outcomes are well labeled, that we're not using billing codes where the information has been degraded permanently. Those big studies that have been designed to measure events have had expert panels actually adjudicating those events. So when you're using machine learning to try and mimic something in the future, some label of outcome or state, the better quality that label is, the better quality the surrogate you can develop. Again, it's about if you have to find if the information's encoded, you have to have a good truth standard for it. So having got the substrate for that and some proofs of concepts, then what evidence is there that if you gave that information to people, could they actually take action? And we don't know much about that yet, but we have some encouraging signs, I think. The first is that this big discovery and delivery platform enables us to deliver more than one thing at once, as you saw a bit earlier. And it enables us to structure products to address a health decision. The one you're looking at here is about diabetes prevention, the situation common in the USA, a middle-aged overweight person with borderline glucose. How do I, if I'm the clinician or if it's me, how do I prevent the development of type 2 diabetes? There's two things you need to know. First of all, how much should I be worried about this issue? And the second is, what should I do? So in structuring a product from a big platform, you can actually have proteomic models for each of the interesting things that might cause you to be concerned. Separate models. Let's say each of these might use 50 or 100 proteins. And then you can also encode proteomic models that are surrogates of each of these other things without having to measure any of those with questionnaires or body composition or even things that people might be lying about. So there are proteomic models that would be reflective of each of these. And these things would tell you what you might do. That if you learn that you're concerned, you can look at these and say, well, if I only could change one thing, which of the things has the most kind of deviant biology? Which of the things in my body right now appears to be most outlandish? Because people can't change everything. And they don't tend to respond to population health-based, well, everyone needs to do more exercise and change their diet. So the other thing this enables people to do is to empirically experiment, the care team or the patient, by changing something and then actually seeing if it changed anything biologically. So this structure of uncovering concerns, finding the biology behind things that they might be able to change, and enabling people to monitor change is a way that we hope will incentivize people to take action. We've only done one feasibility experiment in this vein so far, and it was in a group of people who had high cardiovascular risk, and they were given the cardiovascular risk score that you saw in the JAMA paper and in the circulation paper. And because they believed it, and their care team believed it to be more accurate and precise than the other assessments they had available to, it seemed that degree of accuracy motivated them to change things that they were doing. Now, of course, this is bolder, so maybe they were more receptive than some other areas, but at least this is encouraging. But the key point is we actually don't really know that we can generate all of these insights. We can prove that they're more accurate. We can prove that they're more precise. But until we do something to find, do people really change anything based on these, we don't know if we'll get to any element of the quadruple aim. So what we're doing about that is we're working with the University of Leeds in the UK, part of the National Health Service. We've got a partnership with them to take these products into their patients and measure, do we change their engagement? Do we change their behavior? The first study that we're doing is to take people who are those overweight pre-diabetics, but they're non-compliant with the UK's National Diabetes Prevention Programme. If we present them with more comprehensive information, do we motivate some of those people to change and become compliant? That's the first study. But again, the general point is, until you engage with a health system, you actually can't tell whether better insights lead to any element of the quadruple aim. So I think what I've explained to you is that this paradigm of simultaneous discovery and delivery, having a platform that you can do that while getting the cost down, has some clear advantages, but it also creates some demands, some of which we've addressed, but not all. That you can have actionability if you put together bundles of insights that match a particular decision. There's very early evidence that maybe people will do something that they wouldn't have chosen to do before. Perhaps really importantly is that this is what the initial insights and models that we develop are just the beginning, because the measurement technology always stays the same, and that as we put this out into tens or hundreds of thousands of people, we can upgrade those models with what we learn, and we can add new models without changing any of the measurements. And the risk models that we showed you earlier, they were pretty frugal algorithms, because we used to have the bias towards frugality, because we were going to pull those measurements off the big platform and make them individually. But now, with the 5,000-plex being the delivery platform, you don't have to go for frugal algorithms anymore. You can use some of the deep learning techniques. We don't yet know whether they're going to be any better, but at least we have the option to apply them. We haven't solved all the issues. We haven't yet got down to $5. We're still in the hundreds, but we're aiming to get down to a fraction of that. Currently, when doctors order a diagnostic, they check a box on a form, and that's the thing that gets measured. And that's the thing that gets paid for. That's the thing that's deemed medically necessary. This doesn't really fit that model. And this structure of this new product structure, early evidence is that the clinicians seem to love it. It makes their life easier rather than more difficult, but it'll require education. Well, what will they do? Do they really want recommendations, or are we going to leave it up to them? While we have an initial regulatory path, this is all being run in our CLIA lab, and that fits the regulations. When we start having lots of insights and lots of serious and consequential situations, what's the path to FDA registration? It doesn't fit any of the current analytic validation models. It's more like software, although I think some of the genetic approaches will be a good pro forma for us. Anyway, that's all I have for today, so thank you.
Any questions from the audience? Jim?
So I applaud your vision. I think it's very impressive. As you likely know, so when you do highly multiplex measurements with even gold standard antibodies, you get quite a bit of cross-reactivity with even modest panels. And so I guess I'm wondering, how do you do 5,000? I mean, what's the... Aptamers do have a bit of a checkered history in terms of selectivity and cross-reactivity. And so I'll just ask you to comment on that, and I'll sit down.
Yeah, it's a great question. The answer is they are specific and selective, and the way we achieve that in the assay itself is to exploit a couple of characteristics of the aptamers. One is the slow off rate, that they are selected, the ones we use in the assay are selected to have a slow off rate to their intended cognate protein, whereas the off rate to non-specific binding is much faster. So we use the kinetics to discriminate non-specific binding from specific binding. The other thing we can do is to compete off the non-specific binding by dumping in a lot of cold, unlabeled aptamer to compete off, because they're polyanions. So we can compete off the non-specific binding in that way too. And it's a pretty complex setup, the assay. There's a two-catch system where we first catch the protein complexes, wash away the ones that didn't bind, but then we release those complexes back into solution and we catch them again by the proteins. So there's more than one way of achieving the specificity. And so what evidence have we got that they are specific? Some of the studies that have been published, the big one about to come out is about PQTLs and about genetic protein associations, and I think that's pretty good circumstantial evidence that where they find a PQTL, cis PQTL, it is with the specified named protein. So I think all in all, the performance seems to get better as the plex gets bigger.
Hi, does your assay take into account modifications on proteins like phosphorylation? And if so, does that somehow improve the accuracy of what you're trying to detect?
The answer is yes and no. Remember, I said what we're actually measuring is the availability of a 3D epitope. So if a protein gets phosphorylated and it changes the epitope charge, then the aptamer will bind less well. So that'll appear in the assay like an apparent reduction in concentration, but it'll really be a reduction in that epitope availability. So that will detect that signal, but we actually don't know the origin of the signals. All we know is whether they're consistent and accurate. But the origin could be abundance, shape charge, or competition from a circulating inhibitor.
So really two questions. First one, are you connecting this to the whole genome or not? And second is, using the epitome approach, you had at one stage 5,000 proteins and then 1,300. I'm just wondering whether, when you come down in numbers, the significance of the results go down, and is that due to trying to lower the cost, or is the relationship far more complex than that?
So the genome approach is an interesting one in that a lot of the sample sets that we've gone after don't have genetic information available. And what we're really taking a bet on is that the information that we seek, the biology we seek, is mostly reliably encoded in the proteins alone. Because, of course, the other alternative is to say, if you don't know where the information is, let's measure everything, which is the Aravale approach, which is great. You're going to find the information, but it comes with a cost penalty, and you hurt the most important omic by doing that. So we've taken the bet that there's enough biology encoded in proteins that most of the time, the information you want will beat the best available alternative. So that's the massive bet that we're taking. And then about the relationship between number of significant analytes and plex size, we've gradually built this up from 400 to 650 to 850 to 1,000 to 1129 to 3,600 to 5,000. And it does seem as if the number of analytes involved in a condition does go up as plex size go up. I can't answer yet whether the performance of the model you get starts to tail off. So is there a diminishing return as you increase the plex size? I can't answer that properly because we've kept improving the assay in other ways, and we've gone from having a frugal machine learning approach to now more permissive. So I can't really actually answer it. But in theory, there'll be a time where maybe it's going from 10 to 20,000 that actually you only get 1% more performance because all the biology you want is already encoded in the first 10,000. That'll happen, I just don't know when.
I think this is superb, and I'm not surprised it's complex.
No, ladies first. Ladies first. No.
Okay, so I was just wondering how many from the 5,000 proteins that you measure have already standard clinical actionability? And if not, how many studies, like how long you think it will take until we can change the diagnosis business model, or we can go actually in this high dimensional space where doctors can accept that it's a matter of pattern and not really a single protein?
Well, I think in some ways we make the doctor's life easier. Usually when a new diagnostic test comes along, it's actually annoying because they have a named measurement. They need to learn what the decision threshold for that actually is, and then they need to think, well, what should I do with that piece of information? And what we've done is by training models against things that have labels that they already understand, is it lean muscle mass or is it probability of an event, we actually make life easier because we've already done some of the meaning is encoded within the name that we attribute to the product. So in some ways that makes it easier. And the other thing that makes it easier is what we hope to do is to provide enough insights all at once that the physician doesn't need to go anywhere else. And so it enhances their productivity. We've worked out, how many patient hours and how many clinician hours would it take to get all of the package of insights for prediabetes, for example. It takes about 3 days worth of patient time and about a day of physician time if you were to go and accumulate all of that information from all the other sources. So because it makes their lives easier, we hope that and feedback so far has been that they like it and they find it easier to accept than the old model. But I do agree there's a way to go before we understand, well, how does that translate into a successful business?
So you would say like 10, 20% of your 5,000 proteins are actionable right now?
Oh, I think there are only about 100 FDA-approved protein measurements.
Fantastic, thank you.
Rob?
So just one final question. This is a fantastic resource, but one of the fundamental things is you're measuring all of these proteins in plasma for most of your samples. How many proteins do you think is reliably in plasma that need to be measured? And two is, out of the cohort do you think you've already covered those?
I guess when we get an area under the curve of 1, we'll know that we don't need any more. Of the ones that we do measure in plasma, we know that about 96% of them are signaling in healthy plasma. So the population F statistic, population variance, is higher than the assay noise for about 4,600 proteins. But we'll only be able to answer your question in retrospect when we've done all of these different diseases and we say, are there some proteins that never get chosen? under any circumstances, those proteins never get chosen by machine learning in any of the models. Then I'll be able to answer your question. But going into it, I actually have no idea. And of course, there are some deep learning techniques that always use all the measurements, in which case I have no idea.
So do you think there's 20,000 already in plasma, or is much less than that?
Oh, how many are carrying unique information? Again, I can only answer that in retrospect. When the performance, it was about the other question, when the performance that I need has either reached perfection, and I haven't yet measured all the proteins, or it's reached an asymptote where adding proteins has no longer adds biological information. So I'll be able to tell you in a couple of years, but I can't tell you today.
Thank you.
Okay, then let's thank Stephen again and take a